DIC microscopy is superior to dark field at imaging plasmonic nanoparticles in complex environments. However, DIC also requires greater knowledge and skill to obtain reproducible results. DIC microscopy provides both high lateral resolution and shallow depth of field. Both of these traits are extremely important when monitoring nanoparticles within cells or in complex environments.
To begin, use a scribing pen to place a shallow and short scratch mark onto the center of each glass cover slip, and then clean the glass as described in the accompanying text protocol. Next, use a micropipette to remove 100 microliters of a 0.05 mg per milliliter gold nanoparticle solution from its original storage container and eject the solution into a 1.5 milliliter centrifuge tube. Centrifuge the sample for 10 minutes at 6,000 times gravity. When finished, remove the supernatant with a micropipette to get rid of the excess surfactant from the nanoparticle solution. Next, add 100 microliters of ultrapure water to the centrifuge tube. Briefly vortex the sample to break up the pellet, and then sonicate it for 20 minutes immediately afterwards to fully resuspend the nanoparticle aggregates. Using a micropipette, drop cast 6 microliters of nanoparticle solution onto the cleaned and scratched glass cover slip. Then, carefully flip the cover slip and place it on top of a second, larger piece of microscope glass. The drop should quickly spread out evenly between the two pieces of glass. Take care to avoid getting air bubbles trapped between the two pieces of glass. Use a narrow line of nail polish to seal off the edges of the cover slip to prevent evaporation.